Welcome to the unit on bacterial genetics. Throughout this unit, you're going to hear about things that you've likely learned about in other classes, so part of this should be review. Mainly topics such as DNA replication, transcription, and translation. But we'll also cover some details that are unique to bacteria that you might not have learned in other classes. For me, this is one of my favorite subjects in microbiology, as my training is really as a molecular microbiologist, which means I tend to look at the DNA and the RNA and the proteins inside the cell, as opposed to the overall cell structures. So let's go ahead and get started looking at bacterial genome structures and how those are different than eukaryotes. Everyone should have a basic understanding of the structure of DNA and its role as genetic material from past classes. DNA itself is a double-stranded molecule that consists of two long strands of nucleotides, labeled strand 1 and strand 2 here. The basic structure of a nucleotide has three components. So here is a single nucleotide containing a phosphate group, a deoxyribose sugar, and then a nitrogenous base. The nucleotides are connected by a phosphodiester bond between the phosphate groups and the sugars of the nucleotides seen here along the backbone. It's important to note that nucleotides have a polarity, or a directionality to them. The carbons on the sugar itself are numbered. We've got carbon number 1, 2, 3, 4, and 5. And those numbers correspond to the orientation of the molecule, which means where the 5' carbon is attached to the phosphate is the 5' end of the molecule, and where the 3' carbon on the sugar makes the 3' end. The strands themselves are anti-parallel, so you should notice the 5' end of one molecule lines up with the 3' end of the other strand. The anti-parallel structure allows the nitrogenous bases to line up properly to form the characteristic base pairing of adenine with thymine and guanine with cytosine. You should notice that adenine and guanine have two ring structures, whereas cytosine and thymine only have one ring. The two ring structures are called purines, while the one ring structures are called pyrimidines. A purine will always pair with a pyrimidine, forming three rings spanning the middle of the DNA. This ensures a consistent width of the molecule. In order to function as genetic material, DNA must do three key things. It must store the information that's going to generate all of the metabolic and structural components of the cell, which it does in the sequence of the A's, T's, G's, and C's within the gene regions of the chromosome. It also has to be able to replicate. This allows it to pass information to the next generation of cells. Remember, this is what occurs in bacteria at the start of binary fission, so that there are two copies of the genome for the two daughter cells. We'll look at the mechanism of replication later in this chapter. The third qualification is the ability to mutate. This allows us to generate genetic variation, which means we increase the diversity of a population. Keep in mind that just because two E. coli cells might look the same under the microscope, they're actually just as different from each other as you are from another human, due to differences in the genome because of these mutations. Because bacteria replicate asexually, the majority of the variation that you see between them comes from mutations in the DNA. DNA makes up the genome of all cellular life, but the structure of those genomes are different between bacteria and eukaryotes. The majority of bacteria contain circular genomes, while the majority of eukaryotes have linear pieces of DNA for their genome. The size of the genome, or how many nucleotides it consists of, also varies. Viruses, on average, have the smallest genomes, consisting of a couple thousand to a couple million base pairs, whereas bacteria have more DNA in their genome, ranging from a couple million to about 10 million bases. For example, E. coli has on average 5 million nucleotides in its genome. As we move into the eukaryotes, you'll be able to see there is quite a range in genome sizes. Interestingly, while there's a general increase in genome size from viruses to prokaryotes to eukaryotes, the larger the genome does not actually mean the more complex the organism. For example, humans have 3 billion nucleotides in our genome, while some plants have up to 150 billion base pairs, many times larger than our own. Because eukaryotes have more DNA in their genome, they have more space between the genes of the genome itself, whereas bacterial cells have very densely packed genomes. This means that there's not much extra space in the prokaryotic genomes. The end of one gene is right up against the start of another one. We can see that in this image here. The top is about 50,000 base pairs of a bacterial genome. Each of the green boxes is a different gene, whereas the bottom is just over 40,000 base pairs of a eukaryotic genome. And this is all one gene, with all the little green slivers being the actual gene itself. You can see them here, and here, and here, with the yellow being introns, and then the purple being repeat sequences that used to be called junk DNA, but we now find that it's probably involved in regulation. Keep in mind that the introns, the yellow regions, are not part of the gene itself and will be removed from the RNA transcript before translation occurs, because they don't code for anything as part of the final protein. Remember that genes are just areas of DNA that will be expressed as an RNA product. We often think of this as messenger RNA, but genes also encode for ribosomal RNA and transfer RNA. In eukaryotic cells, each gene must be transcribed into its RNA product individually. They cannot be grouped together. But in prokaryotic genomes, the genes can be organized in an operon, such as the genes A through D that are seen here. An operon is a single series of genes that are all transcribed into one continuous messenger RNA product because they're controlled by a single promoter and regulatory region. We'll talk about those DNA regions in the next couple of videos. The single piece of messenger RNA is then used to translate each of the individual proteins that represent the individual genes of the operon. Remember that bacteria can also contain plasmids, circular pieces of DNA that were not part of the genome itself, but instead confer additional phenotypes or properties to the bacterial species. The process of DNA replication and RNA transcription that we're going to talk about in this chapter have to occur for both the bacterial genome and the bacterial plasmids. This means that most plasmids are replicated similarly to the bacterial genome and that they can be passed to the next generation of cells. A good example of the importance of plasmids is the difference between these two bacillus species that are seen here. This is Bacillus cereus on the left and Bacillus anthracis on the right. Both of these are bacillus species that live in the soil, but Bacillus cereus is not a pathogen, while Bacillus anthracis causes anthrax in humans. The main difference between these two is that Bacillus anthracis contains these two plasmids that encode toxin genes. And it's the additional phenotypes and properties that are given to that Bacillus anthracis bacteria that allow them to be a pathogen if they get inside human cells.